Thank you very much, Claudia, for the very nice and kind introduction. And thank you for inviting me, and thanks to your colleagues for having me here today. I'm delighted to present our new technology to you all. So, as Claudia said, I'm the co-founder and product manager at SiriusXT. So, based in Dublin, Ireland, we've developed a lab-scale soft X-ray microscope for 3D structural imaging of fully vitrified whole cells and bulk tissue samples. So we've a fully functioning microscope offering an imaging service to project partners from our labs in Dublin, and we've just built a fully certified commercial microscope for rollout later this year. So in this talk, I'll give you a brief overview of the technique of soft X-ray tomography itself. I'll introduce our lab-scale microscope, which we call the SXT100. And I'll give you a flavor of some of the scientific use case projects we are currently working on. So why use soft X-ray tomography? This graph here nicely encapsulates the reason. So it's a graph of attenuation length as a function of X-ray energy. So you will see within a certain energy region between about 300 and 550 EV, there is a strong differential contrast between carbon and water. This region of the spectrum is so-called the water window. So basically the contrast in soft X-ray tomograms is entirely natural. So what does this mean? So on the right-hand side here, I'll show a slice from a reconstructed whole cell volume, and you'll notice how the organic matter appears dark against the aqueous background. So in summary, soft X-ray tomography allows the following advantages: 3D structural imaging of whole intact cells without any staining, elemental specific contrast without need for chemical fixation, and cells are in their near native environment. The only preparation step is fast vitrification of the sample. So the layout of a typical soft X-ray microscope consists of a source of some description, a source of soft X-rays, followed by a condenser, which focuses the illumination down onto the sample. The sample itself is rotated over a series of angles, typically in 1 degree steps. And a zone plate, a diffractive element called a zone plate, is used as the imaging objective. So a series of 2D projections are generated, and these are then reconstructed into a 3D volume reconstruction directly from these projections. So the soft X-ray properties allow us to define a bulk slab of biological material. It can be cells or tissues that can be accessed with soft X-ray tomography. So the field of view is about 20 by 20 microns with an X-ray penetration depth of about 10 microns, and resolutions of about 50 nanometers or better can be achieved. So this allows us to address certain application questions that can benefit from soft X-ray tomography, and these are where whole cell information is needed, information on complex networks or volumetric information. Rare events can be nicely captured in the context of a whole cell, as well as compositional information. Most of you will have seen this or some variation of this diagram before, but here I show where soft X-ray tomography sits in the context of other imaging modalities, as well as the size and complexity of biological materials. So cryo-SXT, we like to say, fills the resolution and field of view gap between conventional light and electron-based techniques, and is ideally suited to the imaging of whole cells and organelles. It can also be correlated with light and electron microscopy techniques to investigate molecular interactions within whole cells. So finally, just to put the availability of soft X-ray tomography currently into context, it's currently available at 6 synchrotron beamlines worldwide that are offering the soft X-ray imaging in the so-called water window. One of these is based in the US, there's 2 in Asia and 3 in Europe. And the utility of the technique has been validated at these beamlines, and there's very much growing interest in the technique. So we believe this is a good time to be rolling out a lab-based version. The benefits of a laboratory-based soft X-ray microscope will be on-demand access, as well as a greater scope for development of dedicated correlative workflow techniques that might benefit from soft X-ray capabilities. So now just moving on to the instrument that we have built, the lab-based SXT100, and just showing you a layout here. The microscope has an overall footprint of about 2 by 3 meters. The soft X-rays are generated by a high-power drive laser, and the main modules consist of a source, condenser, and cryo-sample chamber, as well as a CCD camera on a variable magnification rail. So the microscope is, of course, under vacuum and sits on a damped table. The system is, as I said earlier, is fully operational and routinely imaging cryo-prepared biological samples for external project partners. There's a couple of features I'd like to mention at this point. Currently, the microscope accepts samples prepared on standard 3mm EM grids. These are loaded vertically into the microscope via a vacuum load lock, and we're currently in the process of modifying the sample holder to accept autogrids for correlative studies with electron microscopy. Another feature worth mentioning is the standard fluorescent microscope that we've integrated into the sample chamber, which is really useful for in situ screening of the grids and selection of useful cells for X-ray tomography. So this is just an overview of the workflow. This is now fully in place at SiriusXT. We can prepare live cell culture, plate the cells on grids, do fluorescent screening, and then fully vitrify the sample with the option of doing offline cryofluorescent screening if we need to do higher magnification screening. But we have, as I mentioned, the inline microscope, which is already very useful. And then, of course, we generate the soft X-ray projections, and we have data analysis and visualization workflows in place as well. So just an overview of the imaging workflow itself and how it works. First, we build up a fluorescent mosaic of the grid. We identify potential cells of interest. We then do low magnification projection images of selected cell candidates in the soft X-ray with a field of view of about 70 by 70 microns. And if we're still happy with the cell and we want to go for tomography, we'll do high magnification TIL series at about a 20 by 20 micrometer field of view. So acquisition is done over plus or minus 60 degrees with about 60 seconds exposure per angle. We then employ a single script for alignment, reconstruction, and uploading of data to a user folder on Dropbox currently. So within about an hour of data acquisition, the remote customer has the raw data and reconstructed volume associated with a given cell candidate. So just a brief flavor of current scientific projects. We're currently leading an EU-funded project called COSID with 6 partners across Europe to validate the SXT100 in a number of scientific use cases related to viral infection. So I don't have time to go through these in detail, but I'll just give you a brief overview. So this particular study, scientific case number 1, is led by Pablo Gastamenza at CNB Madrid, and he's investigating the effects of hepatitis C infection on hepatocytes. And this study is a continuation of a previous study done at the ALBA synchrotron soft X-ray microscope. So the SXT100 is imaging is confirming the remodeling of membranes and mitochondria in HCV infected cells previously seen at ALBA. And the next step in this project now is to use the SXT100 to look at the reversion of structural damage by direct acting antivirals. In another use case with Nicola Fletcher and Chris Evans at the UCD School of Veterinary Sciences is using the SXT100 to investigate structural differences in genotype 1 and 3 HEV replication in pig and human cell lines, and to investigate the ability of potential therapeutics and inhibitors of the viral life cycle to reverse these structural changes. So the images here shown on the right from the SXT100 show different virtual slices to control cells, and many organelles such as mitochondria, lipid droplets, lysosomes, endosomes, and ER vesicles are, as well as vesicles, are visible in the cytosol. So UCD will shortly move to full-length BSL-3 virus and use correlative imaging modalities to identify localizations such as the sites of replication of the virus and modifications during the viral life cycle. In the third use case, in collaboration with Ralf Bartenschlager at University Hospital Heidelberg, they used FibSim and TEM tomography to characterize the changes to the overall cell architecture of lung epithelial cells upon SARS-CoV-2 infection, and now the SXT100 is being used to confirm these changes on a larger number of cells. So preliminary SXT100 results reveal a reduction in the size and the number of the lipid droplets, as shown here on the right-hand side, as well as crowding of the cytosolic vesicles and elongated mitochondria peripheral to the crowded area. So these analyses are ongoing, and Ralph and his colleagues will shortly move to using the SXT100 to investigate other viruses, as well, for example, the dengue virus. And here with University of Jyväskylä in Finland, SXT100 is being used to investigate for the structural analysis of chromatin and mitochondria in herpes virus infected cells. And here they are using a combination of the SXT100 along with the ALBA soft X-ray beamline. Non-infected cells were imaged in the SXT100, while infected cells were imaged at ALBA. We now have herpes virus infected cells in-house, and these are the next steps for us to actually determine the structure of mitochondria during herpes virus infection. Here's another study done with on cells provided by Maria Harkiolaki from the Diamond B24 microscope. It's a 3-micron thick cell at the cytoplasm and about 5 microns at the nucleus. So on the left-hand side, you can see the raw TIL series. On the panel next to the right, you can see a flipping through the orthoslices of the reconstruction. And so we can observe cristae of the mitochondria, and this has allowed us to make a rough segmentation of the mitochondria. So although in these use cases have shown only mammalian cells, we can also image bacteria and yeast cells too. So we're open and very interested for ideas you may have in terms of more applications of the technique. So another line of study where we have started here is to investigate the integration of soft X-ray tomography into CLEM workflows in what we call TechExchange. So the idea here is that one could combine soft X-ray tomography and electron microscopy techniques with data from fluorescence microscopy to provide a more informative picture by placing basically the functional information into the 3D context of a whole cell mapped at different resolutions. So this is a standard cryo-TEM tomography workflow consisting of cell culture, cryo-correlated microscopy, followed by cryo-focused ion beam milling and cryo-ET. One could insert cryo-SXT here in step 2, along with fluorescent imaging in order to identify regions of interest in a whole cell context. So other workflows are possible as well, such as using freeze substitution and resin embedding after soft X-ray tomography, and then using room temperature TEM. So at the moment, we have 4 projects going, 4 independent projects with 4 different groups looking at different variations of this theme, essentially. Another thing we're doing here is developing a dual grid capillary sample holder facility. So as I said earlier, we are currently accepting standard EM grids. We are now developing the ability to accept cells loaded in ultra-thin walled glass capillaries. This approach would be most suitable for cells in suspension, but could be used potentially for adherent cells as well. The nice thing about capillaries is that it allows 180-degree rotation, thus eliminating the missing wedge of information that comes from limited tilt tomography. So we will use our base cryo stage to facilitate capillaries as well. So it just requires a modification of the sample holder, and users will be allowed to have the capability of using either, or the option of using either grids or capillaries by around the end of March this year. We expect to roll out the capillary function. So also want to mention that we are currently, as I said, operating the microscope from our own labs at SiriusXT, but we plan to move, and we are already in the process of moving to the UCD Conway Euro-BioImaging facility. We should be there by April of this year, and we'll continue to provide the imaging service from there. So we're currently submitting a Euro-BioImaging report based on users to date. So the timeline of the UCD commissioning should nicely coincide, hopefully, with this application. So several potential workflows are available at Conway, such as, in addition to soft X-ray tomography, there'll be correlation of super resolution STED with conventional TEM or SEM, as well as, as I said, ongoing work to integrate soft X-ray tomography into this workflow in the coming months. So we'd love to hear from you in terms of ideas and use cases that you'd like to, case studies that you'd like to try out on the microscope. info@SiriusXT.com is the email address, or contact me directly. We'd love to hear from you. And that's about it. I just want to say a big thanks to all of our project partners. The COSID project is a fantastic collaboration, and all of the other collaborators that have helped us along the way since we spun out the company, and as well as all of the staff at SiriusXT for their great work. So thank you all for listening. I think I'm come in under the time slightly, so time for questions.